If we go back to the very beginning of this chapter, we said that there were two major types of defenses. You have the innate defense and the adaptive defense. The innate was the general defense that you start with, where they throw like phagocytosis, inflammation, fever, natural killer cells. They throw all of these things all at once at the pathogen to try to get rid of it. Now, if the innate system fails and the pathogen is like still alive and in the body, then the body throws what's called the adaptive defense, which is the third line of defense. So this is what we're picking up today. Last class we left off by talking about lymphocytes, and two types of lymphocytes, B and T, right? So today we're going to start with antigens. Now we've heard this word before when we were talking about blood typing. If you remember, the definition of an antigen is anything that causes the immune, or anything that stimulates the immune response. Okay? So, in the case of like blood typing, we were talking about proteins on the surfaces of your red blood cells. In this chapter, we're going to talk about antigens as they are pathogens. So, like your viruses, your bacteria, your protozoa, all the different types of pathogens. Now, again, the word antigen is a general term. Most of the antigens that stimulate a response are big, complex molecules. So we're talking about like your bacteria are full cells, right? Your proteins on the surfaces of your red blood cells, they're like these big structures. There's something called a hapten, which is like an antigen. It stimulates an immune response. But the problem is that the molecule is too small to do it by itself. So it has to bind to something else. An example, the biggest example of this is poison ivy. When you touch a poison ivy plant, the poison ivy molecules themselves are too small to stimulate a response. But what happens is that as soon as you touch that poison ivy plant, the molecules or the poison from the plant combines with proteins in your own body, and the combination of those two molecules, the plant plus yours, together is what stimulates the immune response. Okay, so if those like poison ivy toxins didn't combine with your like body proteins, then you wouldn't stimulate a response because it's too small to do it by itself. Does that make sense? Okay. Now, antigen is what like provokes the response or starts the response. What binds to the antigen is called something called an antigen receptor. So again, the word antigen receptor is just like a general term. When we were talking about blood typing, what bound to the antigen? Antibodies. So in that case, antibodies were the antigen receptor. Okay? So in order to understand how antigens and antibodies work, we have to understand that there's two sides to the story, okay? There's the cell that's being infected, which is you, and then there's the cell that's fighting there's like the bad guys, what's like infecting, right? And then there's the good guys, which is fighting that infection. Now, before we get into all of this antigen presenting cells, let's talk about how the different pathogens affect the different types of cells in our body. If you remember, like last class I played that YouTube video where it showed the different sizes of the different pathogens. What was the size comparison of a virus compared to a bacteria?
No, it's smaller.
The bacteria is bigger, the virus is much, much, much smaller, right? We saw that in comparison to like a red blood cell. If a red blood cell is this big, a virus is the size of the tip of my finger, right? It's very small in comparison. if this is a red blood cell, bacteria could either be a little bit smaller. If this is the size of a red blood cell, the bacteria could be probably like this size, or it could be bigger than the red blood cell, right? So if we start with, like, let's say that this right here is a normal, healthy cell. Okay? So this is an uninfected cell, it's healthy, it's normal, it's going through its everyday processes. So let's think about what happens with a normal, everyday cell, okay? What does the rough ER do in our cell? It makes proteins, okay? So the rough ER makes proteins. What does the Golgi do? It packages the protein, okay? So first we make it, then we package it into something called a vesicle, right? And then what happens usually is that protein, the vesicle fuses with the surface, and now you have a protein on the surface of your cell. You guys remember how there's like proteins on the cell membrane, right? This protein right here that I'm talking about specifically is called MHC1, okay? Now, if you remember, some proteins on the cell surface have molecules attached to it to let your body know that you are you, okay? So this is a normal, healthy cell attached to this MHC protein, or to this protein right here, is going to be an antigen right here, but this is what we call a self antigen. Okay, so MHC is this like little rectangle right here, and this little circle like attached to it is called a self antigen. So let's say that a white blood cell, which is fighting infections, comes here and sees this healthy cell and sees that self antigen. Is this white blood cell going to attack this cell?
No.
It shouldn't. The whole purpose of the self antigen is saying, hey, we are the same. We're friends. Do not attack me. Does that make sense? Okay? So this self antigen says, do not attack me. We are like in the same body. Now, let's look at a cell that's been infected by a virus. Does anyone know how a virus attacks a cell?
It injects the corrupts the cell and multiplies it.
Exactly, but the first thing that has to happen is the virus has to enter the cell. Does that make sense? Okay?